Hi, my name is Jian Zhang. I'm a group leader at the University of Queensland. This is a workshop about polygenic prediction. I break down the topics into 6 short videos. I start with the fundamentals of polygenic score, including intuition about how polygenic prediction works and its limitations. The part 2 is about how to evaluate prediction performance and how to visualize the results and what are the common pitfalls. I then introduced the conventional methods, including clumping, p-value thresholding, and best linear unbiased prediction. We then move to more advanced Bayesian methods. I talk about the Bayes theorem, different priors, posterior inference, and some stats-based methodology. I make another video to explain MCMC sampling, but this will be optional for the course. And at the end, we'll introduce our recent method S-base-RC by incorporating function annotations to improve prediction accuracy. Okay, let's start with the fundamentals of polygenic prediction. These slides draw heavily on work and teaching materials from Naomi Ray, who has been pioneering the development of polygenic risk scores theory and methodology. A polygenic score, or PGS, is designed to predict the genetic components of a complex traits, things like height, BMI, or blood pressure, using genome-wide variation. When the trait is a disease, we usually call it a polygenic risk score, or PRS, because we are interested in the genetic susceptibility to the disease. The key idea is that we are not predicting single genes, we are aggregating information across the whole genome. In humans, this is increasingly relevant for precision medicine and prevention. In animals and plants, this idea is already routine and is known as genomic selection. It has been used in breeding programs for decades. Polygenic prediction is actually not a new technology. Back in 1970s, Henderson introduced the best linear unbiased prediction, or BLUP, which laid the foundation for predicting genetic values using relatedness. Once dense SNP data becomes available, Milweiser et al. In 2001 formally introduced genomic selection and Bayesian methods that are still influential today. In human genetics, the idea of summarizing genetic information into a single score goes back to Land and Thompson in 1990, but it didn't really take off until 2009 when Purcell et al. Applied the PRS to schizophrenia GWAS result. Since then, we've seen rapid methodological development. Bayes R, LD Pratt, PRS-CS, and many others, with ideas flowing back and forth between livestock and human genetics. If you read the literature, you will see many different names: PRS, PGS, multi-SNP score, linear predictor, etc. Most of these terms refer to the same underlying concept. It's a linear combination of genetic variants used to predict the risk of trait. In this course, I mostly use the PGS as a generic term and PRS when the outcome is a disease. Well, polygenic prediction is conducted based on GWAS data. And perhaps one of the most important lessons we learn from GWAS is that complex traits, including common diseases, are very polygenic. There is no single schizophrenia gene or obesity genes. As you can see from the GWAS with increasing sample size, we discover more and more associated variants, each with very small effects. And this is what we call the polygenic architecture. And such an architecture is not unique to one disease. Using a Bayesian methods, which we will talk about that later in the course, we can estimate how much variation are explained by SNPs with small effect, how much variation can be explained by moderate effect, and how much by large effect. The results show that different diseases do have different genetic architectures. For example, autoimmune disease may have fewer variants with larger effects, while psychiatric disorders involves many variants with extremely small effects. But the underlying theme is the same, that many DNA variants contribute to the risk, and most effects are tiny. This is why we need a genome-wide approach. So what does the polygenic architecture mean for an individual? To build intuition, imagine a toy example with 900 DNA polymorphic sites that influence disease risk. If the risk variants each has allele frequency zero point 0.1, then on average, a person would carry about 180 risk variants. This graph shows what the risk profile look like for an average person. We have 30 rows by 30 columns, and each cell is the genotype or allele count for the site. Gray means homozygote of no risk alleles, blue is heterozygote of one risk allele, and red is two risk alleles. In the population, some people carry fewer and some people carry more risk alleles. This variation is what we are going to explore in polygenic prediction. Next, we are going to simulate the genetic profile for disease individuals and healthy individuals. So here we have a group of people who got the disease over lifetime on the top, and on the bottom, these are the individuals who didn't get the disease over lifetime. You may have noticed that there is a lot of variation, but overall, individuals are not dramatically different to each other. We all carry risk variants for essentially all common diseases. This is normal and reflects the biological robustness. On average, people who have developed the disease tend to carry a higher overall burden. But you can also see examples where a diseased individuals carry less risk variance than a healthy individual. This is because non-genetic factors also matter, so the genetic profile is not deterministic in terms of disease development. And importantly, each individual has a unique portfolio of risk alleles. This is why summarizing them into a single score makes sense. Conceptually, if all risk variants had equal effects, then the true polygenic score would simply the count of risk alleles. The variance of this true polygenic score across individuals is what we call the additive genetic variance, and this underlies heritability, which is the proportion of phenotypic variance due to genetic differences. In practice, however, we don't observe all of the causal variants. We only observe the variants on genotyping array or imputed SNPs. This leads to the important distinction between total heritability and SNP-based heritability, which is the fraction of variance captured by measured SNPs. This distinction places a fundamental limit on how well PGS can perform. In reality, different variants also have different effect sizes. So a PGS is a weighted sum of genotypes across SNPs. Each SNPs contribute 0, 1, or 2 copies of the risk alleles multiplied by an estimated effect size from GWAS. The key question in this equation include which SNPs do we want to include and what are their weights. For prediction, we actually don't really need to know the causal variance. The predicted power can come from correlated variance through linkage disequilibrium. And usually after the prediction, we will evaluate the prediction accuracy by calculating the variance explained by the predictors or AUC statistic for disease traits. This is the probability that a case ranks higher than a control based on their PGS. Understanding of the above help us to understand the upper limits of PGS. First, a theoretical upper limit is set by heritability because genetics alone cannot explain more variation than heritability allows. Second, a technical upper limit is set by how much genetic variance is tagged by measured SNPs. Third, since the effect size are not estimated without error, a practical limitation is determined by GWAS sample size and data quality. For example, the technical upper bound for prediction accuracy in schizophrenia is estimated to be about 25% on the liability scale. This is the SNP-based heritability captured by common SNPs with minor frequency greater than 1% in the population, or is upper bounded by the AUC of 0.84. But the observed prediction accuracy is only 11% on the liability scale, or AUC of 0.74 at the moment. As discovery sample size increase and methods improve, prediction accuracy will approach the SNP-based heritability, but will never exceed it. In other words, PGS can never be a highly accurate predictor of the phenotypes, nor deterministic for the disease diagnosis. There is actually a little formula showing that what parameters determine the prediction accuracy. As you can see here, the prediction accuracy can be written as a simple function of the variance captured by the SNPs. So this the SNP-based heritability, H square M, and the error in estimating the SNP effect. That's the C term here. The error term depends on the discovery sample size, the effective number of SNPs being used, and SNP-based heritability. Note that the M here is not the total number of SNPs. Instead, it's the independent SNPs considering linkage disequilibrium. And as we can easily see here, as the sample size increase, C goes to 0, which means the accuracy improves, but only up to the heritability captured by the SNP set. To maximize the prediction accuracy, we want the C to be as small as possible. But as you can imagine, there is a central trade-off about the effective number of SNPs. When we're using more SNPs, that will increase the heritability captured by the SNP set. But it also increase the estimation error because we are fitting more parameters in the model. And on the other hand, when using fewer SNPs, that reduce the noises, but we will miss signals that is not captured by the SNP set. This is really a tug of war or a catch-22 dilemma. How to optimally balance the effective number of SNPs and the heritability captured is an unaddressed challenge to the PGS methods. For example, when we are using the whole genome sequencing data, potentially we will uncover lots of rare causal variants. But whether that could lead to improve the prediction accuracy would eventually depends on how much variation would be captured by those rare causal variants. It's likely to be case by case, depending on the trait. Despite these limitations, can PRS be useful in practice? Well, this Kiran et al. Paper presents a compelling evidence for the PRS as a tool for risk stratification in the population. This landmark paper basically shows that people in the extreme tail of the PRS distribution can have disease risks comparable to rare monogenic mutations. For example, individuals in the top 1% of the cardiovascular PRS had a lifetime risk of greater than 10%. So every one in the 9 individuals in the top 1% PRS being would actually develop the disease. This is equivalent to the risk of carrying single rare genetic variants that can inform changes in the clinical management in the cardiovascular disease. This does not mean PRS are diagnostic, but they can identify high-risk subgroups where targeted prevention may be worthwhile. For common diseases which have known rare variants of large effects, we can combine the PRS with those risk mutations. For example, in breast cancer, about 2% of the breast cancer in women of European ancestry are associated with variants in the BRCA1 gene. And you can see that for the BRCA1 gene carriers, the risk actually has a large variation depending on what are their PRS values. This means PRS can modify risk even in people who are major gene risk gene carriers. So the polygenic background still matters, even when a major gene is present. Importantly, PRS are most useful when combined with other risk predictors. Together, these risk predictors outperform either one individually. For example, a risk prediction for heart disease is already well established in the world, like sex age, smoking status, BMI, family history, and total cholesterol. These are all risk factors. As you can see, none of these individual contributing factors is very useful predictors alone. And because their AUC only slightly higher than zero point 0.5, which is what you expect from a random guess. This is where the PRS is. It's better than each of them alone, but it's worse than the combination of all of them. Well, the best result is achieved by combining all sorts of information. And this could be used to inform prescription of statins and other lifestyle prevention interventions. Another interesting point is that the PRS is better than the family history of heart disease, although both are capturing genetic information. And this is an interesting point and may worth a little bit more discussion. Let's look at this a little bit deeper. Let's think about this commonly asked question. Will people without known family history have high PRS? The answer is maybe yes, maybe not. A common misconception is that high PRS will always align with family history. But in reality, many people with high PRS have known family history due to the random segregation of alleles. This may not be intuitive. So let's look at this, go back to our simulation and look at this example. Um, um, we will simulate a number of children from two healthy individuals, and you will see that important genetic differences can occur within a family as a result of the segregation of alleles from parents to the offspring at meiosis. So this is the results of 50 simulated children. The mean value of these children is 180, the same as the population mean because the parents are healthy individuals. However, there is substantial genetic variation within the family. And actually, it can be shown that the within family genetic variance is as large as half of that, half of the genetic variance of the entire population. And you can also see an individual here who has no family history, but by chance, due to the segregation of alleles, has high genetic risk. So we can come to an important conclusion that a high polygenic risk for disease is most likely a consequence of the genetic sampling of risk alleles. And as PRS become more and more accurate, they were increasingly available to increasingly capable to distinguish risk between individuals within a family, even when their family history is identical. This paper gives a nice review of the major clinical applications of PRS. Briefly, PRS may be useful for population screening in the general population to identify high-risk subgroup, as we have seen in the previous examples. This may be first implemented for diseases where population screening program and prevention health management are already in place, such as breast cancer, heart disease, and common eye disorders. And second, PRS could be used to help diagnosis in ambiguous cases, such as type 1 and type 2 diabetes, when the symptoms are not very specific at the early stage. Third, in the near future, it might be possible that PRS could contribute to treatment choices because you can imagine responses to treatment, including the adverse health outcomes, are likely complex traits. However, in exploring the utility of the PRS in this context, like the choice of the drug treatment, will require large data sets that are not currently available yet. And while clinical risk prediction gets most of the attention, polygenic risk scores, or PGS, are fundamentally a statistical genetic tool. So many applications are found outside medicine. For example, we can use PGS to identify genetic correlations between traits, identify causal effect of genetic factors using family design, to study the genotype by environment, the interaction, and of course, genomic selection in agriculture setting. And the value of the PRS may really lies in once it's justified for one disease and the rest of the disease almost come for free. The genotyping needed for calculating the PRS is now relatively cheap. And once someone is genotyped, polygenic scores can, in principle, be computed for many diseases at essentially very low marginal cost. Even small changes in health outcomes can have a large impact on health economics because polygenic diseases are common diseases of society. In principle, the PRS could be available for an individual for all common diseases from birth. So this cost effectiveness will eventually make incorporation genetic data into risk prediction comes true. And using it routinely in primary care could have significant public health implications. Finally, I would like to highlight a major challenge in PRS application, which is the poor chance ancestry portability. Most GWAS data nowadays are European-centric, but PRS accuracy drops substantially when we apply it across ancestries. A recent study reported that the reduction of the prediction accuracy is almost linearly related to the genetic distance to the European population, when the European population is used as the training population. So what caused this reduction and what's the remedy is an active research area. Briefly, this poor chance ancestry portability may arise from different issues. It may come from the causal variants are different across different populations. And the definition of the phenotype may also different across populations. It may also come from, although the causal variants are the same between populations, but their allele frequencies are different, and they may have different LD correlation patterns or slightly different effect sizes. Recent findings have suggested that it is likely that the causal variants are shared across ancestries, but the reduction of prediction accuracies is mainly because of the differences in allele frequencies and LD structures. And as you can see, the European and East Asian, they have very different LD structures. And these are the key papers supporting these statements. And how to improve cross-ancestry prediction is one of the most important open problems in the field. So in summary, polygenic scores are imperfect but useful genetic predictors. Their accuracy is fundamentally limited by heritability of the traits, SNP set, and the sample size in GWAS. A high PRS is mostly a consequence of genetic sampling. And PRS have the potential to distinguish risk between family members who have the same family history information. And the utility of the PRS has been evaluated in the clinical setting and are often combined with the other predictors to best predict the total disease risk. And usually a very high PRS can indicate immediate utility. The value of the PRS may really lies in this one-off generation of genotypes that can lead to the PRS for multiple conditions almost at negligible cost. It's also important to understand what PRS are not. They are not diagnostic and never will be. They are not absolute risk and do not provide the baseline or time frame for the progression of the disease. And they are not and never will be a standalone predictor for common diseases. They are not equally applicable across populations, at least not yet. And hopefully at the end of the day, you have developed some realistic expectations about what polygenic prediction can and cannot do. If you want a deeper background These papers provide excellent overview of the theory, application, and limitation. All right. Thank you very much for your watching.